Okay, folks, if you can see me, it's only a few more minutes now. I got about, we're supposed to start at 1 o'clock. We'll start at 1 o'clock. We'll be here. Well, it might be a little bit after 1 o'clock. Got to go make a fresh cup of coffee. Be back in just a moment. We're looking forward to showing you the bugs here at Facebook Live at 1 o'clock. It's the Friday Live Bug Show. Wait, here, enjoy some slides. By the way, all of these slides are also available. There's a link over there for there's a link over there for our Redbubble page. That's where you can get all this stuff. Well, I don't know if you want that on a coffee cup right there, but there's a couple of them. The Scorpion Moth has been real popular, and so check out the link. And we're adding to it all the time. And if you have any requests, then you don't want that on a coffee cup. Mini skirt, maybe. You can get mini skirts too. Some of them are of bugs. Look, wait. Ah! Can I go faster? No. I don't know if I can go faster or not. Oh, look, you might want that one on a shower curtain. A couple of bees on your shower curtain. Okay. They're on auto, so I can't do anything about it. Okay, folks, just plugging in the mic here, and then we are going to be... Okay, that was just a... Don't worry about that, guy. We're going to be here in just another couple minutes. It's 1:05. See you in 4 minutes. Got to put this thing under, get it up and clip it on and everything, and then we'll get the whole bug show going. See you in a minute. Looking forward to showing you the bugs. It's the live bug show at Bugman Education on Facebook.com. www.bugs.org. Be there soon. Be there soon. Look at the lovely slides. I'm going to change these slides next week. I know it's been a couple weeks. How far can you go through the show with the sound not working? All right, there you go. Now the sound's working. Sorry, and thank you for that, for chiming in there. And listen, if somebody's watching this, you can comment on this, and I should be able to see it. Oh, look, there's 2 comments, and I should have been looking sooner. But this is why I put the comments on, so that I could see this. Okay. Hey, look, can anyone else hear? I think you're Samantha. I got the sound working. I'm sorry. All right, and I should have been looking sooner. All right. This is all part of my learning curve. Hi, I'm Mark from Bugman Education and Science Shows. Here, look, I'll just go back real quick. You ready? In the studio, this is where we started, right? And then I was telling you about these masks. Okay? I'll put the masks back on now. You ready? I was saying, oh, look over in the comments. So this is what I was saying when I had the masks on. I was saying, oh, look over in the comments over there, and you can even get one of these masks for your very own if you want. And so, look, this one has I was telling you, this is a Viceroy. If you go back, then you can match up the sound. Sorry about that. I out-clevered myself and put the mic on too many. All right, I'm learning. Another reason I wanted to put the mic back on again was because one of the things I was telling you about was the school programs packages that I'm getting ready to work on. And so I've been writing the scripts for them, and we're going to have a set of them for different grade levels. So there'll be like a set of, I know I'm talking while I'm doing this. Look, these are our masks. You saw these earlier but didn't hear me talking about them. If you would have heard me talking about them, you would have heard me say, I know this one at the bottom, those aren't bugs, those are birds, those are woodpeckers. But you can still get them on a mask at Redbubble, and the link is over in the comments, wherever I guess on this screen, the comments are down underneath. And so this is just a little bit of a recap, right? And then I went in and was telling you about this, look at this, the viceroy butterfly, which is a mimic of a monarch, and it mimics monarchs because monarchs taste bad. And I'm going to talk a lot about monarchs today. That was the whole point. And so if you ever see somebody point out or call something a monarch, look for these, where are they? These these black lines right here on its wings. The monarch wings, you'll see, they don't have these. And this is guaranteed, this is a viceroy. And if you didn't see this and it was just flying around, they're a little bit smaller than a monarch, and they fly a lot faster too. And so, all right, so now we should have everything going, right? Now we got the sound working. Somebody say yes if the sound is working. Somebody say, hey, I hear you now, if the sound is working. But I think it is. I can see the little lines going. Sorry about that. I did a whole, I don't know if you were here at the second week, I did the whole show without the sound working and then had to do it again. So I'm glad somebody caught me. Thank you. All right. Now, here's where we were. These are monarch butterfly wings, and they're from 1990. And they were probably lab reared, but you could have gotten them in the field in Hawaii at the same time. And this was what I did when I was an undergrad at the University of Hawaii. I studied these white monarch butterflies. Look at that wing right there. It's white. And that white is not bleached or anything like that. It's a genetic variation on these wings. And so that was what I ended up doing in college. And so I'm going to go on and talk about that in a minute, but since I wasn't talking during the introduction, I thought I was, but since I wasn't talking during the introduction, current bug events, this is why this came on, because there's no current bug events. There was supposed to be a 3-day school thing at a place called, I think it was Freedom Trail. So it was camp day, and we were going to do 3 whole days there, and obviously we can't do school programs now, so we're doing that next year, and we'll be scheduling for next year also. And then also the other current events that are happening is I'm putting together a series of educational online videos that are edited, so you don't have, so I'll know the sound isn't on and it won't go out there. This is live, so that's what happens when it's live. But I've been working on the studio, getting the green screen looking green without me looking too green, and so this has been, here, I'll show you the green screen. You ready? You want to see behind the curtains? Here's the green screen. This is without the green screen. Hi everybody, look at that. That is without the, okay, but you're not supposed to see that. Pay no attention to the man in front of the green screen. All right, there you go. Look, now I'm in front of this. I'll come back to that in a second. But K through 2, 3rd grade through 5th grade, 6th grade through 8th grade. And we're going to have about 5 or 6 videos on each one. And we'll use bugs as our basic content, but it's really about ecology, and it's about nature, and it's about respectful interactions, and respect means look again. And so it'll be very interesting bug programs, but with a K through 8 curriculum. And so you can email me, bugman@bugs.org. I'm Mark from Bugman Education and Science Shows. Or you can just fish around on those links over there. Bugs.org, there's a link to our email, and you can just bugman@bugs.org. Okay, okay, with volume turned up, good. All right, so let's see. I'm going to get reorganized here. Dad says always rehearse, and so I probably should have rehearsed this a little bit more. But the reason I'm doing this show today is because I'm going to talk about the research that I did on monarch butterflies when I was a kid. and I get to preach a little bit too near the end. But look at these butterfly wings. This is live here. And so this is your basic orange monarch butterfly. Let's see if I can get this in focus. Okay, I didn't focus. Ooh, look. And then this is your basic white monarch butterfly. And they're basically the same, except no orange in the white ones. Not much. Sometimes there was more or less, but all right, let's see. This is what butterfly wings look like close up, out of focus. There, look. I think I can get closer a little bit. But I gotta. The cool thing about this is you want a lens that doesn't bother the bugs. And so this one, the further, the more you zoom in, the further away you move from the bugs. Ooh. All right, let's see if I can get that in focus. Close. I know I should be talking right now, but the sound is still on. I'm just playing. I'm just playing with the wings. That's pretty close up. Okay, one more little zap. Nope, that's as close as it gets on the monarch wings. And those are, you know, if you could see those a little bit closer, like under a microscope, this is just a camera, or with my other camera, which I haven't figured out how to hook up yet, but if you could see this under a microscope, those are like scales like you would see on the side of a fish, but they're all different colors and they line up that way to make like a mosaic of color. Color, it turns out, is really important to these animals. I'll tell you a little bit more about that as we go along, but here we go. So, monarch butterflies. Check this out. Check this out. Here, we got the whole life cycle in 3 minutes. Look at this. Ooh. I moved the words down because I got word that sometimes it says live right up there, and so look inside the egg. You can see, I think I can stop it. Can I stop it? Stop. Yes. That is like some kind of little mite running by that's teeny, tiny, itty bitty. And then this is the monarch egg, and this is the side where it's chewing its way out. If you've ever seen baby chickens in an incubator, and they've got and you know they're sitting and you'll see one pecking its way out, and the hole gets bigger and bigger and bigger until it can push its way out. And so I sped this up. If you go to our YouTube page at YouTube/PRMantis, PRMantis, PRMantis. PRMantis is one word. Then you can see extended versions of this quick little video. I made this one really fast, so I could, because nobody wants to watch more than 3 minutes. Although this is going to be hours and hours and hours now that the sound is working. All right, here we go. See that little mite running by? And then you can see this guy chewing inside. Oh, and then he pokes his way out. Look at him wiggle his way out. And this guy is tiny. I mean, you can imagine usually you can find monarch butterfly eggs underneath the leaf near the side, because the female will hang. I might have a photo of it somewhere, but I didn't get it. But female will hold on to the side of the leaf, and then reach her abdomen underneath and lay a single egg. So it's kind of as far as her abdomen can reach underneath the leaf usually. And it's usually only one per plant. She puts down a pheromone, a chemical, that tells the other females that there's already an egg there, and there's typically on the milkweed plants that we have around here, there's not enough leaves for more than one caterpillar to grow its whole life up. And so they usually try to put one on a plant. And then during the more dense times of the season, when there's a lot of butterflies flying around and laying eggs, and it's getting near the end of their life, and so they got to dump the rest of their eggs, a female can lay like about 400 eggs or something. Then sometimes there'll be more than one on a plant, but not usually in Ohio anyway. And that's very different than the way it was when I was in school. So, but look at how cute. And their skin hasn't completely hardened yet, but their head has, because they had to chew their way out. So all of those, all their jaw, you know, there's a chemical in their skin called chitin, CHITIN. It's a protein carbohydrate polysaccharide complex kind of mixture of stuff that cross-links in its shell and makes its skin hard, its exoskeleton hard. You know that a caterpillar doesn't have as hard a skin as a beetle might, and so a beetle just doesn't have as much chitin in it as a beetle doesn't. Caterpillar doesn't have as much chitin. I shouldn't read the comments while I'm working. Okay. A caterpillar has less chitin than a beetle has, and so a beetle has a harder shell. There's a couple other things involved too, but chitin's the main one. And so this one, its shell will get a little bit harder, but I mean, it's still going to be a caterpillar, so it's going to be all mushy and everything, all right? And then it starts eating, and it eats and eats and eats, and actually on that video, the one before that, it eats its eggs. And then when it grows, here's the problem. When an insect grows, its skin doesn't grow, and so when it grows, its skin just gets tighter and tighter and tighter, and then its head splits open, and it sheds its skin. And so it's called ecdysis, right? That's called shedding your skin. And so a caterpillar, but everybody knows it goes egg, larva, pupa, adult, right? Egg, caterpillar, chrysalis, butterfly. But a caterpillar has 4 or 5 stages also. So when it gets too big for its first caterpillar skin, it sheds into its second caterpillar skin, and so on and so forth. And in between each skin, it's called an instar. I-N-S-T-A-R. If I would have had it together more. And on the school ones, we'll have the words. Look, I can put words right over there. It'll say egg, larva, pupa, adult, and it'll tell you that egg, larva, pupa, adult is English, Latin, Latin, English. And I don't know why it's English, Latin, Latin, English, but you say larvae with an E at the A-E at the end, and pupae with an A-E at the end, but it's adults and eggs and adults. And so in one of our school episodes, and maybe some other time, I'll tell you about what the English and the Latin are supposed to be if you did it all in one language, but it's just a weird thing that people in English do. So this larva is going to shed its skin, and so this is right in the middle of it, and it just pushes out, and you can see, you can sort of see in the back. Oh yeah, you can see in the back here where all the old skin is gathering up, and it's just pushing its way out of. Again, this is in excruciating detail on our YouTube page, and then it pulls its little back end out, and I don't like having the words over my face, and then it goes back to eating, and that's what caterpillars do, is eat and eat and eat and eat, and then it starts getting weird because they go to make their pupa, and this is after it shed its skin a bunch of times. And so what it does is it makes a little button of silk right here, and caterpillars make silk like spider web, except they make it out of glands by their mouth. So they got these little silk glands. And actually, when you watch caterpillars walk around, most of the time, they're kind of going like this because they're laying down a trail of silk. And when you're walking on a slippery leaf, if you can lay down a trail of silk, then it'll give you a good foothold in case a bird comes to eat you up or something like that, right? And so with this guy, and so then when they're ready to make their chrysalis, at least a monarch, and these other ones that hang upside down, right? Here's its head over here, right? And so they'll go all the way around, and you can't really see it on this, but they'll go all the way around the surface if it's a branch or the underside of a leaf, or they'll make their chrysalis wherever they can. I'll show you later. They'll make their chrysalis wherever they can when it's time to do it. And then right in the middle, they'll put this little bump of real dense silk. And then that'll be what they walk past it and grab onto it with their back prolegs. They're not real legs, they're fake legs, but they'll grab onto it with their back pseudopods, their fake feet, and then they'll hold on while they're doing all this. And they'll stay like that for, overnight, a bunch of hours. And then when it's time, they'll start pumping. And remember, they're upside down, so they'll start pumping. And just like on the second instar, when it was going from, it was probably third instar to fourth instar, when they shed their skin, their head splits open and it gathers at their back. Well, the same thing happens to the pupa. And I said with these kind because they hang upside down. So then they'll grab it with their back legs and then they'll let go and fall down in a J. And I think those videos are up on the YouTube page too. And so some of them don't hang upside down, but they'll make a little button on a stem and they'll attach their back feet to it. And then they'll attach like one strand on one side, and they'll wrap it around their back and attach it on the other side like the kind of like the kind of utility belt that an electrical guy working on the wires would use, and then they just lean back like this, and with their feet attached to the branch, and they'll lean back with the string holding them up, and then they'll shed their skin that way, and so you'll see the pupa hanging sideways, kind of angled off the side of the branch, and you'll see that a lot. The swallowtails do that, and so it just depends. But it's the same basic principle. You hook your legs onto something, and then you shed your skin. Your head splits open. So now it starts bouncing up and down and gathering all the skin at the top, and then once it straightens out here, the head splits, and it splits right in the back. There's a weakened place there called the ecdysial suture, because that's where it splits for ecdysis. It's the ecdysial suture, and then you'll see it split right there. Boom, and then here's where its face splits open. This is the fast version. So here's where its face splits open, and the interesting thing about this, aside from the fact that the caterpillar's face splits open, is that at this point, this is right at the beginning of the pupa. It hasn't. You're probably used to seeing the pupa as a bluish, purplish thing. It's on the cover shot of our homepage now at Bugman Education on Facebook at Bugman Education. And so, but right when it comes out of its caterpillar skin, the pupal case is not a shell; it's an exoskeleton. So it's a skin too. But while the transformation is taking place, in a lot of insects like this one, the shell gets really hard and it doesn't move. But there are a lot of pupae where the animal still moves a lot. Like with mosquitoes, their pupa swim and they're responsive to light and everything. That was in an earlier episode where I showed that they were responsive to light. And so they got to be active because they're in the water. These guys hang on a branch, and there's a lot of delicate stuff going on in there. Maybe sometime I'll show you some pictures that I didn't take, but somebody did some functional magnetic resonance imaging, some fMRI, of the inside of a chrysalis throughout the process of it of it changing into a butterfly, and you can see all the different organ systems forming there. It's very, 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 very cool modern day technology. But this is older technology. But look, you can already see the butterfly face. These are the butterfly legs, and these things right here. Look, that's its antenna, and here's its other antenna. Look, you can see right there. These are its legs. These strings up here, these are its mouth parts, and they're going to end up rolling into a tube. Its eyes. Right here are its eyes on either side. Not that yellow thing, but next to it, those are its eyes. Right there. And so this is its skin shedding up. I'll make it go here in a minute. And then over here, when you see the side, over here, when you see the side view, you'll be able to see its wings. All right, okay, you ready to go? But look, that's its face. And then it hardens, and you don't realize, okay, and then the other part is that the caterpillar was holding on to that ball of silk with its back pair of legs. Essentially, it's pseudopods, right? And then look at the chrysalis holds on with a stem, pediculum, I think it's called, something. I don't remember the name of it, but I was supposed to look that up beforehand. But technology, okay. So, but that stem is inside the caterpillar skin, and it's got to pull it out and attach it to the ball of silk without falling. And so like the caterpillar skin stays attached to the side of the pupa while this thing comes out, and it's got barbs in the top, and it like on the side of a fish hook, and it drills it into the silk up there on and then it knocks off the old skin. The first time I read about this, somebody described it as if like you were holding onto a cliff with a glove on with one hand, and then you took the glove off and didn't fall off the cliff. Watch. Here it comes. You'll see it come out. Oh, we didn't see it come out. You did, but it was real fast. And then it knocks. Okay, and so then, this is the 3-minute version, so it's going real fast. But then over the next hour or so, the chrysalis hardens and changes to the color that you're used to seeing. And then 10 days later, on the 9th night, it clarifies like that. They digest all the proteins and pigments in it, and then the butterfly comes out. Head first, just like it went in, right? its head splits open and it comes out. And I have this close up on our YouTube page. Really cool videos of it. I was going to put them on here, but they would have taken too long. Oh, look, there's a close up right there of it coming out. Don't put the words on my face. Look it. And there it's pulling its proboscis out, its mouth parts. Yeah, oh, showing off now. Look, that's right when it cracks. That's always the most important, one of the most important parts, right, when it cracks open. This is back shoulder. One season, right at the end of the year, I found 8 eggs all right together on 2 or 3 different plants. And I figured, well, they all got to be siblings, so they were going to go on different, like, real close together. So I just set them all up and shot a bunch of them right in a row. Look, there it's getting its mouth parts already. And then its wings are all folded up, and its abdomen, which is usually sleek, is full of fluids. It's got little claws on its, it's going too fast. All right, it's got little claws on its feet that it holds onto that old chrysalis shell. And then it wiggles around and it pumps fluid from its abdomen through the veins in its wings. And in some butterflies, they maintain some circulation in their wings, and in some it hardens and they're just like scaffolding for the tensile strength of the wings. Right? I don't know what's going to happen. I don't know how fast it's going to happen, but this clear pupil skin right here, this is an important part of the next part of this story. And so kind of remember what that looks like. It's just like this empty shell that looks like it used to be a monarch chrysalis, okay? That the butterfly's hanging on to. Yeah, I figured it would disappear. And then it gets some nectar with that tongue and then goes and flies away. Cool, right? So that's the life cycle of the monarch. I knew you'd like that. All right, well, here's what I wanted to tell you about. When I went to college, I went to, let me come over here. let me stay on the screen. I went to the University of Hawaii, and at the University of Hawaii, I'm going to make me bigger. You ready? At the University of Hawaii, I studied monarch butterflies. And what I studied was, hang on. And what I studied was, hopefully this will work. Yeah, okay. What I studied was monarch butterflies. Well, I didn't expect I was going to study monarch butterflies when I went out there. And so here's what happened. This is what I knew. Well, I'd always collected monarch butterflies, and some of that video was from before I went to school, which was in 1987 was when I went to Hawaii. And so this was pretty much what I and almost everybody knew about monarch butterflies, was when the overwintering sites were discovered in Mexico. And it was a big National Geographic front page. And I have this because it was a slide from my talk. What I'm about to show you is a bunch of slides from my talk when I got graduated from my undergrad at the University of Hawaii. And I graduated in 1990. So all of these slides were shot in 1990. And as I was getting ready to, and these butterfly wings that are behind these slides right here, that are on the Sony cam right here, look on the close-up on the Buggo Vision cam, these wings are from back then too. They're 30 years old. And they have stood the test of time, right? Stood the test of time. Now, the slideshow that I did a lot of the slides on a computer. And back then, it was really hard to do. I mean, like, just to do text and to get a blue background and all that, it was ridiculous. Like, things that would take 20 seconds now on something like PowerPoint or whatever you use for that kind of object handling. I mean, it would take hours just to make some of these slides. I'll show you in a minute. But, so this was one of the slides. This was one of the slides in my talk. The slides are perfect. I've had them in a little folder, and they, where does this end? Yeah. So I had them in a little folder and in sheets, and they're like brand new. The butterfly wings are like brand new. Now, the files that I used for my slideshow have been gone for, even if I found them, they're probably like on those, I don't know, oops, sorry. I don't know how much computering stuff you remember, but back then, to save your stuff, it started off on like a big black disc like this that was 640K or something, and then we got these little blue ones that were littler and hard, and so you called them floppy, but they were hard, and that was 1.4 megabytes, which is 1.4 megabytes. That isn't even one of these photos, and so, but that's how we started, and everything was hard, but so cool because you could do it, right? And so I had a light table at the university that I could use, and my old slide camera that shot, 35 millimeter slides. I still have the camera, and it works just fine also. And I've had so many cameras since then that don't, but back in the old days, man, stuff worked. And so, I would put this on the light table and make a slide out of it so that when I did my talk, I could do a slideshow. Sort of like I'm doing now, except for it was so different back then than it is now. All right. This is what everybody knew. The monarchs migrate to Mexico. They gather in huge populations. They're so cool to raise. I raised a whole bunch of them since I was a kid. And I never exhaustively collected them, but if I found a caterpillar, a lot of times if you find a caterpillar and you know what it eats and it's from around there and you can easily get a lot of what it eats, you can just raise it and then let it go. I wouldn't do it with a whole bunch of them. I'll come back to that, but I wouldn't do it with a whole bunch of them, but it's great to see, and it's super great to show kids. And so teachers who want to show this to kids sometimes will order painted lady butterflies that are raised in a lab or mass raised, and then they tell them to release them in the nature. You don't buy stuff and release it in nature. It's a great thing for kids to see. So if you buy painted lady butterflies, that's great. Just don't release them, and explain to kids that if you do release them, the worst thing that'll happen is they'll live happily ever after and have lots of babies. And you don't want a whole bunch of purchased and released animals out in nature as a general rule of thumb. Okay. So that's right. So anyway, I went to Hawaii. And then, when I was in Hawaii, my first day on campus, and I was just walking around, and this was one of the things I saw. I figured I'd have to blow some of these up. Look, these are slides, literally, Kodalux slides. And so I left the slide things on here because they're so charming, but I can blow this up so you can see it a little better. I figured I'd have to play around with these things a little bit. That, you still can't see it, right? Because it's so little. But that right there is a caterpillar. Look, it's right, well, I'm pointing with I can point it with my finger. There's a little caterpillar walking across a walkway, and look, there's somebody with their slippers on. In Hawaii, nobody wore shoes. They always wore flip-flops, but they called them slippers, right? They call them slippers, whatever. They didn't say the R. But, so this was walking in front of the zoology building. Not the first day I was there, but one day in the same place. And it's a big front to the building with windows and stuff. And on one of the windows, somebody posted a little caution caterpillar crossing sign here. And when I got to Hawaii, I was green. I had no idea about any of it. I didn't know that there were monarch butterflies there. I thought while I was there for part of the time that I had seen I thought I saw a monarch flying on the other side of the island, but I couldn't tell for sure. And but then my first day on campus, and I'm in front of the zoology building, and this walkway, I got to the end of the walkway, and on the wall at the end of the walkway was a poster, behind glass, and it was about white monarch butterflies in Hawaii. And I was like, monarch butterflies in Hawaii? White monarch butterflies in Hawaii? Oh, and the other thing is that this poster was on it was under an overhang. Like the walkway was under an overhang that was 2 stories up. And so it was this real high overhang. And if you looked up on the overhang, that empty pupil case, the one that looked like a little piece of empty cellophane or something, there was 100 of them. I had never seen a pupa monarch pupa in nature. I was in my late 20s. I had never seen a monarch pupa in nature, and I had never seen an empty pupil case in nature. And there was 100 of them up there. And then I get to the end of the walkway, and there's a sign that says "White monarch butterflies in Hawaii," and I'm like, monarch butterflies in Hawaii, white monarch butterflies. I got to do something about this. So I looked down at the bottom of the poster, and it said Dr. John Stimson, room 306, this building. And it was my first day on campus, and I had made a pledge to myself that I was going to say yes to every opportunity I had. And so I went right upstairs and knocked on Dr. Stimson's office and said, I want to study monarch butterflies. And I'm probably paraphrasing this and not remembering it right, but this was a really formative time in my understanding of how science and how research works. I said, I want to study monarch butterflies. And he said, great. We study monarch butterflies here. What's the question? And I was like, what's the question? What's this guy talking about, right? And I said, how can I study monarch butterflies? And he said, it's easy. What's the question? And I was like, aren't monarch butterflies cool? I didn't know what he was talking about. The question, what are you talking about? The question. And so what he was talking about was that you don't study monarch butterflies. You study interesting questions in ecology or development or evolution or whatever genetics, whatever you're going to study, and then you find the animals that best fit that question. That's how I ended up studying mosquitoes in grad school. Monarchs were a lot more fun, but they were both equally as interesting. And so Dr. Stimson ended up explaining that over the years. It took a long time for me to internalize the fact that it's about an interesting question and not so much about an interesting animal. But everybody's like, ooh, I want to study dolphins. I'm going to be a marine biologist. But you're sitting on the boat for hours and hours and hours and hours and hours, and you're not studying dolphins, because being a marine biologist is a lot. But you can learn interesting things studying dolphins if you have the right question. All right. So this question was about white monarch butterflies, and I mentioned the plants that the monarchs live on here, and you've probably seen these before. And this is I didn't get a picture of the full plant, but you're probably familiar with it's called Asclepias syriaca, because Linnaeus, when he named it, thought it was originally from Syria, but it's really native to this part of the United States. At least the eastern part of the United States, and so they're very common here in Ohio. You'll see fields and fields of them, and actually there's been a big movement to replant milkweed plants, and they get the big, huge, fuzzy, pubescent, fuzzy leaves, and then they get these big seed pods with all the flying seeds with the little fuzzy umbrella. And so that's probably what you're used to. A single stalk with a certain amount of leaves on it. It's going to grow, it's going to make beautiful purple flowers that smell great, excellent pollinator flowers, and then it's going to die, make seeds and die, and then grow next year. So it's perennial. Now, in Hawaii, another thing I didn't know about the monarch butterflies is their host plant is in the same group as the milkweeds, but it's, oh, look, there's a white one. Look at how beautiful. Look at how beautiful. That's a white one. And somewhere I had one that had our tags on it, but this one doesn't. But when I'll tell you a little bit about the research we did, but we would put a little mark on their wing up in this cell right here. Believe it or not, with a Sharpie, and I actually collected one of our butterflies 8 months later on the other side of the island. And so I don't know if the Sharpie was hurting it or not, but that one managed to live for a remarkably long time, which was pretty uncommon out there. Let me make sure this is lined up right. Okay. I know that kind of stuff bothers me. You guys probably don't even notice, but it bothers me. All right. So here, this is the plant though. Look, it's got, if you're familiar with milkweeds, the leaves are almost exactly the same shape. They're a little bit bigger, a little bit wider. And then, but they're fuzzy still, pubescent is what that's called. And then they get a very similar flower. This is called a crown flower, a giant crown flower. And it's in a different genus, but it's in the same family, the Asclepiads, and it's in the genus Calotropis. It's Calotropis gigantea, and it's originally a South American plant. Okay, so monarchs eat this. They typically only eat plants in the Asclepiad family because they make a chemical on their skin, in the plant, that was originally supposed to stop caterpillars from eating the plant. Plants and caterpillars are in a big war all the time anyway, and so plants make tons of, there's all kinds of stuff that plants do that going on that we don't even know about. But they make like latex stuff that'll gum up the caterpillar's mouth, and then they make real supposedly bitter tannins. And then these have this stuff called cardiac glycosides in them, which are toxic. Although the caterpillars learned how to eat them and not die from them. And then, because if you eat them and die from them, you can't learn. But they learned how to eat them and not die from them, and then incorporate those toxins into their skin. And so if a bird tries to eat a monarch butterfly or a monarch caterpillar, it throws up. And then birds aren't very smart, so it will eat another monarch caterpillar and it'll throw up. And birds aren't very smart, but eventually they learn, don't eat black and yellow and white striped caterpillars. Don't eat orange and black striped butterflies. Don't eat those guys right there. Those black and orange stripes are warnings, and when a bird eats enough of them and it remembers you throw up every single time, it'll stop eating them, and this is called aposematic, A-P-O-S-E-M-A-T-I-C, aposematic coloration. Apo is away from, automatically away from, when you see that warning color associated with throwing up. You might notice in this picture, those do not have a warning color. And in most places, black and white is not automatically a warning color. It can be, because birds learn. And so it can be, but it isn't automatically. But this black and orange, kind of universal. Red and orange, black and orange, black and yellow. Those are the warning colors. School bus, yield sign, stop sign, right? Okay. Well, I wanted you to, we're supposed to see these things. Forgot. Okay. Oh, not that one. Okay. That's all right. We'll get to the next ones. That's not supposed to start over every time. All right. There you go. So the crown flower, instead of being a single plant, is a bush. It's like lilac bush size. They get huge. This is a stand of about 5 of them. And the reason, and then there's more down there, and this is also the reason why there were so many pupae on the overhang, because where that walkway was, and then the building is over here, and the white monarch butterfly poster is at the end of the walkway. To the right was this big stand of, my hand is running off this, and then was this big stand of crown flower plants. This is all news to me. I had no idea. And by the way, it was in the fall, because I was starting school, right? And so that's good, because in the fall is when these guys are active. Monarchs are active in the fall. And that's another unusual part of it, because in the summer, the plants have leaves, but there are no butterflies to be found anywhere. And so these crown flowers, wherever there's a stand of crown flowers, there were monarch butterflies everywhere. And so you could stand at these plants and you could see the whole life cycle. You could see eggs hatching. You could see that whole 3-minute thing. You could see caterpillars shedding their skin. You could see caterpillars hanging upside down in the J, going into a pupa. You'd usually have to look off the plant to find them going into a pupa. And then you'd see like maybe 10 butterflies flying around these plants, and you'd see white ones and orange ones flying around these plants. You'd see mating pairs of monarchs, which you almost never see in nature here, because they're almost solitary here, because the plants are individual weeds spread around, right? Here, there's these big bushes, and it's like this, this big singles meet up here, right? Come on out to the crown flower plants. And so it was a different way of living, and you could even see different behavioral things, which I recognize here now with the monarchs here that I never noticed before. But the males will patrol around these plants, and if they see anything moving, they'll dive bomb on the female. Well, they'll dive bomb on it, whatever it is. Sometimes I've seen them go after birds before, because they'll go after anything moving and then wait till they get close to see if it's a butterfly or not. And then if it's a female butterfly, or sometimes if it's a male butterfly, they'll knock it to the ground and wrestle with it and try to mate. But a lot of times the female flies away. She ends up getting the final choice anyway. Okay, so during the year, these guys will completely defoliate their crown flower plants. These are the same ones. So this is this is the beginning of the season. I know it's a different angle. This is the beginning of the season. And then this is like after the first wave. And so the monarchs show up like in October. They show up. At this time, this was in the late 80s, nobody knew where they were coming from or if they were going anywhere or what happened, but they would just show up after being gone all summer. And the plants would look like this when they showed up. And then they just, it was a frenzy. And then in like a couple weeks, the plants would be completely defoliated. Now, I say completely defoliated, but if you look closely enough, some of these plants still have leaves on them. Little teeny tiny leaves, right? And so, so they're not completely defoliated. Never mind, it won't get big enough. But they're not completely defoliated, but they're mostly defoliated, right? Just mostly dead. They need something to live for. And what they live for are for the little teeny tiny leaves on there that are just starting to slowly grow back as the other ones are getting finished off. And then the monarchs will come and lay eggs on those. And so then they get completely defoliated a second time. And then the monarchs disappear. And as the leaves grow back, nothing eats them. And then all summer long, nothing eats them. So that is something we didn't study. But what we did study was the white monarchs. And so the question was, why are there white monarch butterflies in Hawaii and there aren't white monarch butterflies on the mainland? Now, some of you who are watching might know that, yes, there are some white monarch butterflies on the mainland, and I'll come back to that in a minute. Look, this is a very rare picture of an orange and a white flying together. I didn't say it was a good picture, and this is a slide also. When I found these slides, my objective, I had some of them scanned already, but there were other ones that I'm using in this that I didn't have before. And so my objective was to get the slides, and I've got this scanner. It's hard to find a scanner that will do slides because it's got a light from above and below. And so I got a scanner at a thrift store for like $6. It worked great once I downloaded the software for it. But then I got forced into using Microsoft 10, Windows 10, and it was horrible. And so now I can't use that scanner anymore. So these ancient slides that are 30 years old, I ended up getting one of my movie lights that has like a diffuser on it, and I put the slide on top of it and then use my cell phone to take some of these photos. So that's new technology. And these files that I have, I won't be able to keep for very long because technology is going to change. It'll all go to the cloud, blah, 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 blah. Who knows where it's going to go? But I'm going to have these slides forever, and they're already 30 years old, and they look great. They still look like they're brand new. Of course, find a slide projector for them. You got to go back to the thrift store. Okay. So, look, there was no difference between the monarchs. The caterpillars were exactly the same. You couldn't tell the difference between the orange and the white until late in the pupa. Sometimes when it started to clarify, you could tell, but it wasn't always. You couldn't always be sure. They're the same butterfly. And so, if you cross, it was just like Mendel said. If you cross two whites, you get all white offspring. It's a homozygous recessive trait. So if you put two of the white alleles together, all the babies are white. But orange is dominant. And so if you put an orange and a white together, then it depends what the numbers are going to be, but they'll be mostly orange or all orange. And then if you put two oranges together, they might be all orange or they might be something in between. Actually, if you put an orange and a white together, yeah, some of them could be white too, because this orange one can carry a white, but it won't show up. But this is because they got two of each color, essentially. They got an orange and a white, an orange and a white, or a white and a white, or an orange and an orange. Those are your only choices. And so if you're going to be white, you got to have a white and a white, right? If you're going to be orange, you can have two oranges or an orange and a white, and you'll still look orange. Okay, so I don't know which one this one is, but that is two whites. So anyway, knowing those genetics and understanding a little bit about what was going on in the field, because Dr. Stimson had done this before, we did some research on it. And by the way, I mentioned that they had been seen other places, and they have been seen other places. Now, I did this in the late 80s, like I said, and at that point, it wasn't so easy to do literature research. I remember I ended up having a bunch of advisors because the zoology department was in one college at the University of Hawaii. It was in the College of Arts and Sciences, I think. But the entomology department, which was where I was enrolled, was in the College of Tropical Agriculture and Human Resources because it was part of tropical agriculture. And so I needed to have an advisor from my college, CTAR, College of Tropical Agriculture and Human Resources, and that was Dr. Johnson, and Dr. Marshall Johnson. And actually, I contacted him to try to do a little guest thing with him, but we couldn't get the whole thing set up right. And he wanted to talk about parasitoids and, the biology of leaf miner parasites and all that. And I'm like, I just want to talk about butterflies and how fun it was working with him because he was my undergraduate advisor officially. And he did things like took me into the basement at the science library. You go back through all these big stacks, and then there's encyclopedic versions of this thing called biological abstracts. You can still look for it online, and I think they've got them online in index. But back then, every year or every half a year, I think it was every year maybe, they would put out this big, huge index. It might have been every month. And it was an index that was this big. And all it would have would be entries of all of the biological scientific papers that had been put out in that month or however long it was. And you would go through and look for it. And then associated with that big, huge volume would be a whole series of other volumes that were from the time period. And you would look up the code for it and then go and find it in these other books. And then you would get the abstract, which is just a summary of the actual paper. And if you needed the paper, then you'd have to go talk to the librarian and get them to do an interlibrary loan because they didn't have the books, so you had to go find it, and then they would usually have to pay for it to get it from. It was crazy. Now, you just Google things. And so I was thinking about if anybody knows any young researchers, the way things have changed is interesting in two ways, because now if you want to go do research, all you got to do is go do research, go Google stuff, right? Google Scholar is almost as good as Biological Abstracts was, and it's a whole lot easier because you don't have to go. It was crazy. It was crazy. But Dr. Johnson was a really good undergraduate advisor, and I'll never forget, that's another formative lesson, like talking to Dr. Stimson, who was my other advisor, who said, "What's the question?" That was important, and it was a good thing for me to learn and remember. And then Dr. Johnson taking me downstairs and showing me the biological abstracts, and this is how you do research. You go find other papers of research that have been done, and if you're going to make a claim in your paper, you better cite it where you got that information, or you better have it in your own results. Or just don't say it, or say it's speculative, or qualify it somehow, right? And so now when I'm on Facebook, I yell a lot because people will just say stuff, but you got to find out if you learned it somehow, it came from somewhere, and that should be part of you sharing the knowledge, right? Okay, so on biological abstracts, I found these 5 sightings starting in 1896, and this was in 1986, 88, okay? And so in almost 100 years, 5 had been seen, and 3 of them were on the United States mainland, and all before 1921. Look at that. Before 1922, right? So they're very, very rare, but they were here, but very rare. And so what happens, at least in theory, what happens is that if an allele, that copy, that white version of the butterfly wing, if the white version is at a really, 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 really small proportion in the population, 2 whites aren't going to get put together very often. And so the chances of coming up with one that looks white, because it's 2 whites together, are really slim. You might get 1 in a billion, however many. I don't know what the numbers are. But in a huge population, eventually, oranges that have an orange and a white each are going to mate. And when oranges that are orange and white, you only see the orange, but they can pass on that white. And if both of them pass on that white, the offspring will be white. but birds won't recognize it. And so if it's one in a gazillion and a bird sees it, it doesn't think, ooh, that's one of those orange ones that makes me throw up. A bird sees it and says, ooh, look, a white butterfly, I'm going to go eat it. And it doesn't matter if it throws up because it doesn't encounter enough white ones to teach it not to eat those butterflies. That's what aposematic coloration is, or aposematic, or however you say it, right? That's what that means, that it's a warning color associated with something bad. Now, if the oranges didn't make you throw up, then you eat them. Whites don't make you throw up, you eat them. But if the whites start making you throw up, you don't eat them. So here's what was happening in Hawaii, all right? Here's what was happening. And by the way, okay, I'll tell you while I'm thinking about it. The reason that I qualified not seeing many in the United States now is because recently, since the big scare when the numbers of migrating monarchs in Mexico went way, 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 way down, this is an attachment for the what you can do part that's coming up at the end. But one of the things that happened in response to what was supposedly a crisis in the overwintering sites in Mexico, one whose cause has still not been completely identified, was that everybody got very nervous about the monarch butterfly populations, and they started trying to help them. And the way they started helping them, there were a lot of different ways. And one way was planting more milkweed plants, which is a great way to help them. Milkweeds are native. They're constrained by their habitat. You find native seeds, you move them to someplace else. A lot of the native milkweeds have been knocked down because they like disturbed areas. And so around farm fields and stuff, they would grow around farm fields, and then when Roundup started getting used and they could make genetically modified corn plants that wouldn't get killed by Roundup, or whatever, soy plants that wouldn't get killed by Roundup, then they could spray Roundup as much as they wanted and it would kill all the weeds, but it wouldn't necessarily kill the crop. But it was a terrible thing for the monarchs. And then also, for the milkweeds, and in turn for the monarchs. And then also when depending on the timing of when they mow, the medians and berms, and I am a berm man after all, and the berms and medians on the highway, milkweeds love that too. But if they mow it too soon, then it does real damage to the monarch butterflies. So controlling that kind of stuff were some of the there were big campaigns to get them to mow later in the season or earlier in the season, before or after the main monarch part. And the milkweeds are resilient; they'll grow back. But if they're not there when the monarchs are there, or if you mow them when the monarchs are caterpillars, then you're hurting the monarch population. So they've been working on that. And also, people have been building monarch way stations that have milkweeds all over the place, and all good stuff. Another thing that started happening is people started putting in milkweed gardens and then going out and getting all the eggs and bringing them inside and protecting them, understandably protecting them, so that birds wouldn't get them, or ants wouldn't get ants. I've seen ants take eggs off of plants before and knock little caterpillars off of plants before, and so they would protect them. And when then they would raise them up inside or in a covered porch or in a cage or something, and then let them go. And they would let go like 300 adults at one time. 300 adults that were grown outside of nature, it's almost like a laboratory where they're not subjected to natural selection. And even though it won't necessarily impact wing color, there's other differences that I don't know if anybody's ever measured, that I don't even know if you could measure, between a white and an orange, that who knows? Who knows what impact it has. But when people started crossing a bunch of butterflies artificially and raising the offspring artificially and then releasing them, more whites have shown up on the mainland in the United States. There was a lady in Hawaii, and I think I even heard of someone in Virginia or someplace, who was actually looking for the whites when they got them so that they could cross them and get all white offspring, and then, woo, look, I can sell these. And then all of a sudden there's all these whites here, and they don't belong here. They're as bad as Chinese mantises, even though they don't eat hummingbirds. They don't belong here either. So when I started hearing about a bunch of white not a bunch, but a few sightings on the mainland of white monarch butterflies, I was like, that's not right. Something's wrong there. Excuse me. So, okay. That was not the virus, by the way. That was talking too much. Let me get a fresh beverage. Hang on a second. Look at these beautiful butterflies. Okay. You have to cover your coffee here because of the fruit flies. So, what we did was we went out in the field. This was the title. This is my slide. Look, this is a slide. This is the title slide to my talk. I don't know if I can make it big enough here. Color variation in monarch butterfly wings on Oahu, Hawaii. Evolution of a species on an island. Island biodiversity is a really interesting thing. It's like a little laboratory. And so I heard somebody talking about this whole lockdown quarantine situation as being an interesting laboratory study in nature, because you don't usually get this many people off the streets and this much manufacturing not being done. And so people are measuring sort of the pollutants in the air and how much it's affecting it. And it's a very complicated picture, but you don't usually get an opportunity like this with like a small fraction of the traffic on the road. Well, you also don't get a chance to study evolution in an organism like a monarch unless it's isolated on an island, or unless a bunch of people start releasing them uncontrolled, whatever. I'll try to quit complaining about that. You ready? All right. So these slides were from my talk, right? So this you had to do on, I think it was a program called Harvard Graphics or something, and you make that, and everything back then was white letters on a blue background, and I was like, ooh, look, I can make pictures, and a lot of the professors didn't like that at all because it was old school, and so now you got TED Talks and everything, and you got live bug programs like this where I can do videos and whatever. All right. This got longer than I expected because I was doing it without sound for a while. So if you're still here, hi. But I'll probably, I don't know, who knows. All right. So, and this is one of the caterpillars. And so one of the things that we did was we went out, or we didn't actually go out and collect caterpillars because caterpillars are a lot of work. But what we would do is we would do some false crosses. Well, not false crosses, but like, you hold a boy and a girl up their back ends, their abdomens together, and they're going to mate. And so you could take two whites and put them together if you want and get all white offspring. And then we use these cages here to raise the caterpillars in. And I would go around the island and steal as many milkweed plants as I could, or crownflower plants as I could. And it got real hard. I was competing with the wild monarchs, and they would eat. If you have 100 caterpillars, they'd eat a lot. They were like zombies. They'd be like, we need leaves. Do you have any leaves? Give me leaves. And I'm like, no. And then I'd have to go run over to the girls' school that was next door to the university and steal some from their plants. And it was like I don't know what school it was, but I snuck in and snuck out, and I don't think I ever got seen. And so, but sometimes you'd go, like it'd be a weekend, and the caterpillars would run out of the leaves, and you think these things are secure and stuff, but man, hungry caterpillars, they would get all over the lab. And so you'd come in like on a Monday, and there'd be pupas hanging from your stack of papers, and look, here's their old caterpillar skins they knocked off. And so these were pupal cases. This is just a stack of computer paper. I think it's dot matrix with the little lines along the side. And then this is actually underneath the printer. This is hanging underneath our printer in the lab. So there were pupas all over the place. And in nature, they're all over the place too. And so we would use these for lab crosses to reinforce our understanding of the genetics of it. Is it true that if you cross all whites, you get all white offspring? The answer, based on our studies, was yes. Okay. And so then the other thing we would do is we would go out and exhaustively collect pupae. And so this site in front of the zoology building, and by the way, Dr. Stimson studied shallow coral reef biology professionally, but he just started getting interested in this because the crown flowers were out in front of his building, and in back of there was a stand in the front and a stand in the back, and they were everywhere. And so he's like, well, this is cool. I'm going to start studying it, and he released a bunch of papers on it. And he did some very, very interesting research on it. some of which I helped. Okay, this is the part I helped with. And so the other part that we would do is we would go out and exhaustively collect the pupae, and then and when I say exhaustively, they were everywhere. So this was like on a table under like a makeshift bench or something, and they're hanging all under there because there were some crownflower plants back there. And so wherever there were crownflower plants, if you look around during the winter months, the rainy season, there were pupae all over the place. So we would go collect them. And if you on the top of that where they had that button of silk, if you real gently just grab up there once it's hardened, you can just pull, the whole button of silk will come down. You can just pull that whole thing down, and you can hold it up by this mat of silk that where the button is in the middle. And then we would take a piece of tape. Well, oh look, they were everywhere. That one was actually at our study site most of one summer, and you couldn't really get it off of here because it was wrapped too tightly around there, so I just left it on there. But the other ones we would take, and we would take chicken wire. This is a piece of chicken wire, and we would fold it so it made like a little roof thing, and then we would take a piece of masking tape, and you could write the location and date on it, and then hang those things. They would fit right through the chicken wire, and then when the butterflies came out, they would hang down. Those down there are still drying, right? And so we did hundreds of pupae this way. And on that overhang that was 2 stories up, I couldn't reach that, and so I made a pupa scooper. I got a big long stick, like some kind of metal pole. I don't remember what it was from, but it would expand, and then I took a plastic fork and kind of bent it back and taped it to the top. And I had a butterfly net, and I would get up there with the plastic fork, and I would get above where the pupa was hung on with the plastic fork, and I would gently pull it down, and it would come sailing down, and then I would catch it in the butterfly net. And that's how I collected a bunch of our pupas from the 2-story overhang up above at the zoology building. But we would collect them from all over, and we would note where they came from. And the idea was to collect all of the pupae we could after they made it to the butterfly. I mean, after they made it past the caterpillar. The caterpillars were out there getting selected on, and you don't know on what measure they're getting picked off. And so whether or not it had anything to do with they had the white gene or not, who knows? But you let them go through their life cycle out there in selection, and then we would collect the pupas, which technically were out there, could be selected for also. And I've seen them getting eaten before, but it didn't seem to be as big a factor. Okay, and also we would just randomly collect these guys, and they were easy to collect because they were just sitting there. And so then we would see what proportion of white monarchs came out. I don't know if that's this slide or not. Let's see. Oh, this is actually a little bit ahead, but the idea here was we would see what the proportion of white monarchs came out of this. And then these were just wild caught. So we would put a little mark on them where they came from and what the date was, and we had a little code for that. So it was like three little numbers or letters on that little cell in their wing with a Sharpie, and then we would let them go. And they seemed to do fine when we let them go. And so then we would go back to those sites periodically and randomly collect as many as we possibly could and see what the proportion of white was that we collected, allowing for a lot of potential errors, like whites are harder to see, and they seem to behave the same. The difference in behavior was between males and females. Males would patrol around the plants, and females would furtively hide in the leaves, laying eggs and trying not to get attacked and knocked to the ground by the males. So what we ended up thinking was going on, and probably going into this, what Dr. Stimson was thinking was going on, was these birds here. And I don't know if this will get bigger or not, but you can sort of see right in there is a red-whiskered bulbul bird, B-U-L-B-U-L. It's pink. It's pink. Oh, my camera might have just gone off. I got to do a battery here. You know what? Enjoy these pretty slides here. I'll show you a picture of the bird while I do. I got to put my battery back in. Look at me frozen there. Maybe I can make me disappear. But if I do, my mic might go off. Nope, can't do anything right now. So I'm just going to, I'll move this down so you can look at the beautiful bird. And this is a bulbul bird, Pink Nenotus Pycnonotus jocosus. Pycnonotus jocosus, and I'm going to do this while I'm putting in a new battery in my camera. I got to buy a cord. I know, a professional bug person would have a cord for his camera, but I do have a charged battery. So while you're looking at that beautiful picture and me frozen on the screen, I should be able to stick this battery in here and get everything back going again. Ready? You ready? This should have gotten us back up and running. Let's just make sure, okay? Hopefully that'll get us back up and running. Are we running? Hey everybody, look, here we are. All right, I just have to do one little fix here because I moved it. You would think, don't you have a tripod? And I would say, dude, you cannot imagine what we've been going through in this green screen lab to make it work well. Look, there, now it's all lined up and everybody's happy. I'm happy. You happy? All right, so that is a red whiskered bulbul. Where were we? That is a red whiskered bulbul, and it's called that for because look right here, it's got these little red things on its side. It also has a red vent, and but the other there were two bulbul birds, two species. This one was probably look how beautiful it is though, introduced from South America, and then the other was the red vented bulbul. And just like the red-bellied woodpecker is a red-bellied woodpecker, but really it's got a red head, but it's got a little blush of red underneath. This red vent right here, don't let that fool you. It's this little, look at that right there. That is a red whiskered bulbul. I always get them confused. And then this is a red vented bulbul, and you can barely even see its vent right there, but it's got a red patch underneath its vent and no whiskers. And look, it just looks like a more evil. These guys are so smart. I had a friend who raised one from when it was just a fledgling, and it would talk and eat out of her hand, and she wouldn't want to take it out of the cage till it went to the. She had it in a cage because she raised it from when it was a baby, and so she would sit there and say, "Make doo doo, make doo doo, make doo doo." So it would poop, and it did it before she got it out of the cage, and then it would poop, and she would take it out of the cage, and it would prance back and forth on its perch and say, "Make doo doo, and then it would go to the bathroom, so you would take it out of its cage. But really smart, and I've seen them team hunt monarchs before. Bugs. Like I saw two of them sitting in a tree. One flew out and grabbed the beetle and then dropped it in midair, and the other one flew out and grabbed it, and then they flew off together. And so they'll hunt together and everything. Very, very, very smart bird, introduced from South America. Introduced from South America in the late 60s. Which was about the time white monarch butterflies started showing up in any kind of numbers in Hawaii. And so from when we collected the pupas and let them go in the field, this is sort of the scenario that we were postulating in the research that I did. And this was like 3 years worth of research that I did. And I had a huge data set, well, it's not huge by today's standards, but back then it was like almost 400 individuals with a whole bunch of observations on each one. And so there were big long columns, and there was this program called DBase or something that would crash all the time, and I'd have to re-enter all the data. And I learned save your work. Once you do so much work that you don't want to do it again, save your work, right? But if we released these 7 butterflies out into nature from our random collections in the field, our exhausted collections in the field, 5 of them were orange and 2 of them were white, so that's the starting proportion. And then we go back and collect again in the field, and we find a different proportion because birds start to eat them. Where is the other bird picture? Because birds start to eat them. And so so for some reason, the bird picture isn't showing up. I think I have to erase some of these, okay? I'm just going to erase some of these real quick. I've never done 20 slides before, for good reason. And so now maybe this will release it. Release the slides. Yeah, now it's blank. Okay, so never mind. The other slides won't go now. But All right, here, enjoy these beautiful monarch pictures while I redo the slideshow. All right, you ready? I'm just going to do this real quick. By the way, one of the reasons these shows are free is exactly this, because it's part of my learning curve. So I hope you enjoy this while I'm flailing trying to figure out what to do next. This is what to do next. All right. I guess a slideshow can only have 20 slides in it, maybe, because that was 20. All right, let's see if this shows up now. Okay, see if this works. Where did we leave off? We left off right. We saw all these, right? I know. This is not professional. This is how it goes. Look, there, we saw that. Look, we saw that. This is the big one. Look. So, as I was saying, folks, if you go out there and you only collect 5 and 3 of them are orange, but 2 of them are white, well, this proportion is very different than this one. This is 2 out of 7. Do the math. This is 2 out of 5 because the birds preferentially eat the orange ones. That was our theory, that you put this many out there, and instead of the orange, these bulbul birds can eat monarch butterflies without throwing up. So the orange ones aren't protected. Theoretically, the white ones aren't protected either, but they're harder to see against the leaves when they're down there. They're harder to see against a cloudy sky, against a blue sky. Look, they're even harder to see against a blue sky. And so now, if the birds, this was one place where Dr. Stimson and I kind of disagreed, but if the birds can eat the orange ones, then they'll start looking for the orange ones. They won't even pay. They'll be like, oh, that's a white bottle, but look at that orange one. Ooh, I'm going to eat that one. So it might have turned into actually an eat here sign. What used to be a warning, this thing will make you throw up, now could be an eat here sign. And that was something that we never did enough research to find out, but we did find a lot of circumstantial research that this was going on. Another data point we would do was we would collect wings around the site, because as you can see on this beautiful illustration, the birds would drop the wings, and they would take the caterpillars, and I've seen them eat the caterpillars too, and they would bang them against a wire or something and knock a bunch of green stuff out of them before they would eat them. So they had ways to kind of reduce, if I remember right, a lot of the glycosides, the toxins in here are concentrated in the wings, and so they would knock those off and just eat the body, and they would very aggressively eat monarchs. So that changed the whole situation, because they were on an island. Everything was an introduced species. You put it all in this little laboratory and you see what happens. And that was the research that I did. And it was really interesting. And so I learned a bunch. And one of the things I learned is that if you do artificial crosses like this one, you don't release them. This is you cross, I don't know what we crossed here, but it could have been it could have been an orange and a white, and a bunch of oranges came out. Normally with an orange and a white, at least one of them would have been white. With 2 oranges, sometimes one of them might be white. This could have been 2 oranges and none of them were white. But when you do a bunch of artificial crosses in the lab, and you're trying to keep track of the proportion of a certain color in the field, you don't just let all these go. Like, ooh, I'm going to mate 2 whites and I'll get 100 baby whites, and then look, I'll just let all these go out in the field, and then I'll go out and count the whites that are in the field. And so that was something I learned, and one of the reasons that it alerts me and makes me nervous when I see a bunch of people releasing 300 adult monarchs or 200, and they should be, because it's kind of a good thing, but it's probably the wrong way to save the monarchs. That's what I'm guessing, because you just pollute the gene pool out there that doesn't get selected in the caterpillar. So another thing I learned about this was really interesting, and I graduated. These slides were from my final talk, but here was another interesting thing I found while I was doing this. If you look at these 2 arrays of female monarchs, I'll make them a little bit bigger so that you can see what I'm talking about. But in the oranges and the whites, they would show a spectrum of dark colors on their wings. And so if you look at this one on the bottom here, this is what a male looks like, but that is not a male. These are all females. But this is pretty much what a male looks like. So you would see some females that look almost exactly like males. But then there was like this whole spectrum. And you would see some that were almost completely black. They look like a whole different species. Look at this. I don't know if it's getting bigger or not. Okay, look at that. It's almost completely black. And especially the black scales would concentrate near their thorax. And anybody who's still here and who has paid attention to the shows last week and the week before, I mentioned that has to do with thermoregulation, with warming up. And it happened in the whites and the oranges, but only in the females. I don't think I put... Oh, no, that is a male. I'm sorry. This one at the bottom is a male. Wait. That one's a... The one at the bottom here is a female, but this one's a male. I think. It could be that they're both males, but some of the females, like I think this bottom one's a female and the top one's a male. And then on the oranges, those both on the right are males, okay? But then look at these females, and look at how dark they get. And it also seemed to be accompanied with smaller sizes, and sometimes in the lab this would happen, where they would be smaller, sometimes really small, and sometimes really dark, and those things seem to be associated. I started collecting data on it, but then I graduated and I wasn't getting paid for any of this. But this is still a very interesting study about these monarch butterflies. And the theory that I was thinking about, well, look, once you get eaten, and it doesn't matter anymore, and there isn't this really strong selective pressure to be bright orange. If you don't look like this on the mainland, you're going to get eaten. But a bird on the mainland recognizes this. But in Hawaii, they might not, they'll be able to eat it whether they recognize it or not. And so if a bulbul bird can eat you, then it doesn't matter what color your wings are, right? So all of these things that monarchs sacrificed a long, long time ago in genetic history to be bright orange now have a chance to re-express themselves. Maybe. Who knows? But a lot of the other butterflies in this family are white and black. Some with a little bit of orange on them. The closest related to a monarch have orange on them, but some of them are white and black. And there are other just white and black butterflies. And so that might have been something that monarchs gave up in their gene pool by it becoming so rare because they always got eaten that they're just all orange. And now you put them in an island laboratory and start changing the conditions and add a non-native host plant and add, which by the way is on the low end of toxicity, so it's not as poisonous as some of the other ones, and then you add a predator that doesn't eat them anywhere else apparently, and then all of a sudden all these things start happening, and if they don't have to be white anymore, then, and this didn't happen in the males stayed completely clean, but the females would get really dark, which theoretically would allow them to warm up earlier in the morning and get out and start laying eggs and stay out later in the evening and lay eggs longer than the males are active, because that's going to be temperature dependent even in Hawaii. And so the darker you are, the more sun you can absorb and the more eggs you lay, but also the more you can avoid having the males around, because whenever the males are around, they attack the females whenever they see them, they knock them to the ground, and even if they're already mated and won't mate again, it interrupts the whole, they're busy laying eggs, they don't want to get messed with. And so, who knows? Who knows what was going on there? But it was something else interesting that happened. Another interesting question that came up while I was answering this other question, and maybe monarchs aren't the best animal to study it on. One of the papers I came across that I pulled but I didn't bring in here, but I still have the reprint that I had back in the 80s, the original reprint that I got of a paper by Joel Kingsolver, who was at the University of Washington in Seattle then, and he studied a species of the white butterflies you see flying around all over the place, cabbage whites, and they have black markings on their wings. Next time you see one, look at them because they vary in the amount of black that they have near their thorax, and they have a black thorax. And when they land, they sun themselves. And he studied a population up the side of a mountain. And as you go up a mountain and you get higher altitude, then the average temperature goes down, right? It gets colder the higher you go up the mountain. And he found that as you go up the mountain, the populations were getting more and more dark scales in their wings. So cool. Same species, but as they changed habitat up the side of a mountain, you could see a physical change in the genes, in the versions of the gene combinations that were surviving. More dark scales on your wings. You could warm up faster when the sun came out. And I think something similar might have been happening here, but I don't know. I was just a kid. I was just a kid who ended up graduating doing this research, which, obviously you can't see that because it's off screen right now. You ready? This was not my graduation picture. This was supposed to be my graduation picture. Not in here. But, and I don't, it's not that easy to add a picture. But listen, I'm going to admit there were a few flaws on this particular show because I spent a lot of time redoing the green screen. The green screen is really tricky because I'm getting my picture. You ready? No. The green screen is tricky because it doesn't look like because to get it to be green, I don't know where this picture is, but to get it to be an actual green screen, you have to this wasn't even on here. Okay, to get it to be an actual green screen, you have to make it a whole different color. I had the hardest time with the green screen, and so I got it real fixed up now. So I kind of like the way it's working now. And so look, this was me graduating. I'll put it behind me. There. This was at my graduation. Since I studied monarchs, but I was always a big mantis fan, and I had this balsa wood praying mantis. Shoot. That I can't get a hold of here. I'm sorry. This is a lot more difficult than you think it is. I know I make it look easy. It's a lot more difficult than you think it is. Look, this was on B1 of the local newspaper, and this was me getting my diploma. I got my graduation picture in the newspaper, and then because I had this mantis on my hat. This is me. And that's the school president. And underneath these words here, he's handing me my diploma. And we're shaking hands, and he's laughing because I had that thing on my mortar, and I only rubber banded the legs on because I didn't know how to affix it right. And so as I would move around, I could feel it slipping and starting to fall apart and stuff. And I leaned over, and I said, yeah, you should be smiling. He was smiling at it, and I'm like, yeah, this thing could fall off here at any time. And everybody started laughing at me, and that's when they got that picture. But this research was really valuable for me because as the only undergrad in the department in Hawaii, I had access to all grad students and all professors, and they were all really nice and really great and super, super helpful. And I learned a lot of what a grad student would learn. And even though it was just a Bachelor of Science degree when I graduated, I ended up getting into a doctorate degree, a PhD degree in Massachusetts on the weight of this work because I never officially got a master's degree. I got a teaching certification instead. And so this was a really good and fortunate thing for me to have experienced when I was in Hawaii. I was really lucky. And I thank Dr. Johnson and I thank Dr. Stimson. Those were my two main advisors. And then I think Bruce Tabashnik was also on my committee. We had really, really great researchers at the department back then in the late 80s. And also I got to work with a researcher named Jay Rosenheim on different stuff who taught me so much about how to do science. And so nowadays, when I hear about kids wanting to go into science, I just want to tell them about this stuff and how to do science and how cool it is and how you end up getting 300 observations and then you don't save it, and then you got to re-enter them all. And sometimes it's hard, and sometimes it's a nightmare. But if there's something you really like monarch butterflies, or like the evolution of color in butterfly wings, then I encourage you to go out and find somebody who's doing what you like, and then you can do it. I'm going to try to add one more image, which I know is probably a big mistake, but I'll try to, because I didn't have time to do this at the beginning. This has been a process putting this whole thing together, and so we'll have to see if it'll do this one more image. But in the meantime, this is the big finish. So in the meantime, I hope you all enjoyed looking at the bugs today, and I hope you enjoyed my funny little stories about bugs today. And so this won't load up by itself. And so maybe it will. I don't know. I should either be doing this or talking, but not both, right? What I was looking for, the last thing I was looking for, was a link to our science paper, and I didn't find it, so don't worry. But if we're still on here, I lost everything. But if we're still on here, the reason that we're doing this is to show people bugs, and I'm a little bit nervous now because I don't see myself or any of the stuff, and so I'm afraid that we might have gotten lost somewhere along the line. Let's try a different screen. And so maybe I'm on, maybe I'm off. I don't know. Nobody knows anymore. If we're still on, thanks for checking out the bug program. It was so fun showing you the bugs today, talking about my research from Hawaii. And I don't know what I'm going to say now because I don't know if you can still see me. But if you can still see me, contact me at bugs.org, bugs.org, about our school programs that we're doing. We're going to do a couple of canned programs that libraries can use for this summer. We've had a couple requests for it. You can go to Redbubble and find all of our products that we have. If you still see this, they should be listed over there on the comments. Leave comments, ask questions, tell stories, and we'll interact with you online. In the meantime, if you're still watching, thanks for checking out the bug program today. It's been a real blast showing you the bugs, and we'll do it again next week, Friday at 1 o'clock on the live bug show. We're looking, oh look, there I am. Now I can see me. We're looking forward to showing you the bugs. Next week we will. In the meantime, enjoy some beautiful slides.